No, of the microphone. Then we can't see you.
Okay.
So what's the problem?
Shut up. Don't be a meanie.
Or do. Are you guys all settled in and ready? Have you got your drinks? Are you tucked in? Are you comfortable? Very slow. And I'll begin.
Now this is a story all about her.
Alright. Hi! For those of you who don't know, my name is Libby. I'm in my second year of a Bachelor of Genetics here at the ANU, and I'm here to talk to you about genetics and the X-Men. The alternative title I was going to go with was, what? No, why? No, that really doesn't work. Okay, to begin with, I want to tell you I'm no expert on either genetics or the X-Men, despite the fact I'm studying one and I'm enthused about the other. So if you want to pipe up with anything correcting me on either genetics or the X-Men, or just want to put in your input, feel free to raise your hand and I'll call you. Just don't be like, this is bad and you should feel bad, because then
She'll cry on you.
I will cry on you.
She'll feel bad. Cry.
And you will not like that. I also want to clarify, I'm not referring to the X-Men movies, I'm referring to the X-Men universe. Because when I looked at the movies and looked at all the science in those movies, it would have just been 45 minutes of me screaming and ranting, which, while entertaining for you, is not really entertaining for me. Because I just had bugbears about the whole series of movies. This is assuming I have a tiny bit of knowledge from my previous genetics XSAC. I will do a quick recap and go over some important things. But again, if you have questions about genetics and want me to clarify anything, do tell me, because that's what I'm here for. Okay, so a quick, very, very information-packed recap. You certainly don't have to absorb all this. I'm happy to remind you. Basic sort of dogma about genetics is that DNA turns into RNA, which turns into a protein. DNA is this thing here. You've got a sugar phosphate backbone, which are connecting what we call base pairs, which is adenine to thymine and guanine to cytosine. RNA is kind of single-stranded version of DNA, except instead of thymine, it's replaced with uracil, which is a different base pair altogether. It has kind of similar shape, but somewhat different functions. RNA is then passed through what's called a ribosome. A ribosome is basically a two-part protein that clamps onto it, reads it 3 base pairs at a time. Those 3 base pairs are known as a codon, and those codons correspond to a different amino acid, which is very tiny, but the code corresponds to this. And once those amino acids chain together, they fold into a protein. And that's basically all DNA's expression all the time. It's just DNA to protein, and those proteins form different molecular pathways to form different tissue structures, or to signal out different things, or promote a physical response from other cells. Okay, so types of mutations. You can either have mutations of the sequence, also known as point mutations. You can have sequences of the chromosome. Point mutations can include insertion of a base pairing in the sequence, a deletion of a base pair in the sequence, a substitution where one base pair is swapped for another, and combining those together can either result in missense, where if you were to swap one, it would code for a different amino acid, or nonsense, where if you swap it, it codes for a stop codon. So if you can see here, there are about 3 codons, three sets of 3 that'll cause translation of the RNA to protein to stop altogether. And that's known as nonsense, so it will stop prematurely. To add to that, there's also a frame shift mutation. Remember when I said that 3 base pairs correspond to a protein? It's usually quite accurate in determining where exactly to start, but mutations can skew that up entirely. So if there was a deletion mutation, it would be okay reading up to it, but then there would be a frame shift to the left to the corresponding DNA sequence, and so you get a entirely different set of amino acids as a result. Chromosomal mutations, you can get a lot of different things actually. Chromosomes are kind of fickle and they usually happen during cell replication. One of the easiest ones I can describe is translocation, where a bit of one chromosome lops off and attaches itself to a bit of another one. That's the one I have. I have
It's like a 2-5?
a 2-5 or a 4-5 mutation that's balanced, so I haven't lost any genetic data that's apparently vital, which is great.
Sure.
I'm very sure my dad also has it.
It's actually where it's recombined, it's created the STIRP gene.
Yeah. Occasionally throughout the night, you might hear me burst into Shojin, but it's nothing. Yeah. So yeah, apparently completely harmless. I'm not sure if I'm a mutant yet. I haven't received any information, but I'm okay. Over here, you also have chromosomal fusion here, where it's a byproduct of cell replication that chromosomes can kind of fuse together, and you can also get genome duplication, which is also attributed to
I just want to say, chromosome fusion is the main reason we know the difference between us and apes.
Yes.
Because apes have 48 chromosomes, we have 46 because 2 of our fused.
Yes.
That's the only difference.
And then there's genome duplication. It's also known as polyploidy or duoploidy, where you can get copies of the same chromosome, and it's triploidy 23 is down.
Yeah, I think 21. 21.
Thank you. Triploidy 21 is Down syndrome. Okay. Any questions before I continue? Does everyone understand me, or are you just looking blankly, being like, I just heard Latin backwards? Okay.
We think we can.
Okay. If you have any questions, feel free to interrupt. Yes?
Should we be worried that the Sentinels from X-Men are going to come and take you away?
Pretty amazing.
No, because that would be amazing
I
to
would
watch.
be a really useless superhero. I'd probably try to hit someone and get hurt in the process.
She's like a reverse Wolverine.
Yeah. It's true. Okay. So, mutation procedures. Nothing like what you see in the X-Men or almost any other scientific top movie. I'm pretty sure that's a given. As you can see, there's this representation of movie science montage and actual science montage. It's way too true. Like, even if this isn't for genetics or anything forensic or mutation related, it is very true. And if you just, so you see all this sciencey stuff with lasers, and the difference is the movie science montage, it says, paint flecks from the killer's clothing match an antimatter factory in cell grade. Let's go! And then the actual science montage says, okay, we've determined there's neither barium nor radium in this sample. Probably. And that's about as accurate as mutation procedures go in real-life applications, particularly if you're working from scratch. Over here, I have a slide that I may have taken from one of my course materials this semester, but this is a very simplified view of transforming a bacterium and trying to express a specific protein in bacteria. Before I continue on, bacteria are way different to the cells within our cells. The procedure is very different because we don't have these things called plasmids, which is very, very crucial in terms of bacterial transformation. Okay, so assuming you're starting from scratch, there'd be way more steps before the bacterial culture, in which you take a culture of cells, and in our case, we irradiate it under ultraviolet light, and then we see what happens. We select and screen those irradiated cells for something that we think will be useful. We will extract that DNA, put it into a plasmid, and amplify it, so we've got a decent amount of DNA to work with. Then we'll try and insert it into into a fresh culture, and usually how we do that is we shock bacterial cells, because what happens if they're shocked either by extreme heat or cold, or through electricity, which is known as electroporation, is that when the cell membrane breaks, bacteria will basically scramble for any piece of DNA to find, because it needs that DNA, and at that point, any DNA could possibly be helpful. And what happens is we call those confident cells. Biotech companies will usually produce these confident cells in batches, and they'll give them to you. They'll have them frozen, and they'll give them to you, so you just give them DNA and hope for the best. So, assuming that the bacterial cultures take up this new DNA, you then regrow it, reselect, and screen it. And usually we use not only antibiotics to kill off cells that typically pick up the plasmid, because the plasmid, and you can't really see in this picture because it's blurry, but among regulatory sequences and the DNA that you want, will usually be an antibiotic resistance gene in there. Anyway, you grow it on a selected medium so that the bacterial cells that have taken up the plasmid survive. Then you screen those survived cells to see if any took up the plasmid correctly and express the mutation you're after. That has a lot of steps. Anything can go wrong between those steps. Absolutely anything. And it's so much more difficult to do the same with animal cells, eukaryotic cells, because when our cells are shocked with extreme heat and electroporation, there's much higher chance of our cells lysing, as in just bursting completely, and our cells don't have that mechanism to take up whatever DNA can find. We've got to, well, I'll show you in the next one, but we've got to sort of force the DNA into it. Anything you wanted to add, or not?
No, I think we're all good.
Okay, any questions before I continue?
Nope.
Okay. Okay, so, mutating a body is even more difficult than mutating a cell, and that's because cells can't really transmit and pass along DNA or any sort of genetic information between each other. It just doesn't quite work.
At least not in eukaryotes.
Yeah, not in eukaryotes. But for us, it's so difficult. There are some sort of sketchy ways in which you can sort of tell DNA at people and see what sticks, and I'm not using that phrase as a figurative one. You literally okay, a gene gun is a thing. It is a thing. Basically, you load up a gene gun with tiny, tiny, tiny nanoparticles of metal that have the specific DNA that you want to insert into the cell attached to it, and then you aim it at something, aim it at an organism and fire, and hope that cells will take it up.
It has like a 1 in a billion chance of working.
Yeah, it's sort of just it's not there yet.
But you're dealing with like 2 billion cells at a time,
so yeah.
Two of them will work.
Two of them will work, and you don't have any control over, apart from where you point the gun. You don't really have much in the way of control as to what cells take it up, and if the cells incorporate the DNA into the correct location. So you might get something similar to chimerism or mosaicism, where certain traits are just expressed in random locations. Sort of like freckles, but absolutely not like freckles. It just looks like sort of a freckly kind of pattern, particularly when you're dealing with something so small. And that's known as bioballistics, which I learned today. It actually has a term, bioballistics, and that's the coolest thing I have discovered today. It's such a cool word. But
Horrifying concept.
Yeah, I know. On a similar vein to metal nanoparticles, they're trying to incorporate them into food or digestible substances to try and have an ingestible gene therapy. It's still in sort of production. They've tried it in animals, and there's been a very, 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 very low success rate, but it might be more useful to get genetic information to the endothelial cells, so the cells covering the inside intestinal or digestive system that's sort of trapped in the body, instead of, trying to get a gene gun down there and firing it open for the best. Which, that's a horrible. That's
whole
feel
new
bad
kind of
when
food
you-
products.
No, what?
Ignore me.
Okay, I will. Okay, I've also put in engineered vector, but I should have made that question mark bigger because they're trying to have retroviruses and adenoviruses that will actually go out and try and infect the cell with genetic information. But it's still very much in progress because not only do you have to evade the immune response and the immune system, but it has the same problems with metal nanoparticles in the sense that you cannot control where that DNA ends up being spliced in the genome. It might end up that it gets spliced in a very, very crucial part of cell regulation, and you could have a cancerous mutation come up, which I'll come to in a bit. No, let me clarify, not one mutation, one single mutation will cause cancer. Cancer is basically a accumulation of different mutations that sort of come together and have all these sort of characteristics of immortal cells, which is a very, very bad term to use, but it's not one mutation that causes cancer. It's a bunch of mutations. Sorry, I'm sort of going off on these tangents. Have I got everyone here?
It's all the good tangents, though.
Yeah, all the good tangents, but I want to make sure people still understand. Yes? No? Yes? Okay.
What about mutations at birth? Like mutations in reproductive cells? What do they do?
Well, if they work, and if... Hang on, can you define mutations? Like natural
So we're
mutations
shooting
are part of...
Well, like
the
if someone was to take like
More
embryonic,
at your...
can I talk? If someone was to like take embryonic cells and mutate those and say they were viable, would that mean that the mutation would be in the entire organism? Or?
If it's literally a single embryonic stem cell before it becomes a zygote, theoretically, yes. But if you were to clone an organism, one of the main things they do is they take they take something blast.
Osteoblast.
Yeah.
No, not osteoblast.
Yeah, that's what osteoblast create bone.
Yeah. Blastocele.
Blastocyst.
Blastocyst. Yeah,
Okay.
that thing.
They culture a zygote, they let it grow to become a blastocyst. They'll usually
Blastocyst is how many cells?
It's like between 16 and a whole bunch.
yeah, between 16 and a whole bunch. Sort of, it's a massive sort of just conglomerate. It's like if you were to take a sphere of frog spawn, that sort of dimension and that sort of amount.
Lots.
Yeah, a lot. It's between 16 and a whole bunch. And they'll usually try and, well, one method, of course, is they'd usually try and insert the DNA into part of the blastocyst. And if it was engineered like that, then it would probably exhibit chimerism or mosaicism, where it would only affect specific portions of the body. Because at that point, Hox genes, which I explained in my last XSAC, will be in place. And Hox genes are basically the regulators as to limbs go here, torso go here, head goes up there, you have 2 legs, that sort of thing.
And which way bustle goes.
And which
Very
way bustle
important.
is very, very important. Surprisingly, not surprisingly. Anyway, okay. But if it was just one egg cell, one egg / one, actually, one egg or one sperm that undergoes a mutation, it's a matter of, is that mutation a recessive or a dominant allele, and will it get expressed? Did I answer that, and do people understand me, or am I just rambling?
Makes sense.
Okay, good. I'm doing better than I thought I would. Hooray! Okay, on to the actual subject. I'm going to be looking at 3 different case studies from 3 different characters in the X-Men universe. One of them you would probably agree is in the X-Men, but 2 of them, in fact all of them technically are. Wolverine and Maggot were part of the X-Men. Deadpool is part of the Uncanny X-Men, which is a bit of a spin-off, but it's still part of the X-Men universe, and you'll see why when I come to him. But we're first going to start off with good old Wolverine. and just, no, it doesn't
There's
quite
too much muscle.
work. Okay, so I had to do a lot of hunting to get a cohesive backstory on him, because part of me doesn't even think that the writers of X-Men know his backstory, because there was a bit where he was like, he was raised by wolves. No, Sabretooth was his father. No, it was all just a dream. No, they were engineered. No, they were engineered memories. And then there was finally a book a comic that was supposed to, and I'll say supposed to illustrate his backstory once and for all. So this is what I'm going on, because I don't know, being raised by wolves doesn't really explain a lot. Neither does actually Sabretooth being his father. Anyway, okay, so Wolverine was originally born as James Howlett. He was born very frail and very sick, and he remained that way. He was in a bit of a dysfunctional family. His brother, John Jr. Howlett, died at a very young age. His mother went into a mental facility out of grief. And later down the track, Thomas Logan, who was the groundskeeper's son and James Howlett's best friend, Thomas Logan was the father of his best friend, killed James Howlett's father, and Wolverine unfortunately had to walk in on this situation as he saw the groundskeeper kill his father, and then he sort of went into this blind fit of anguish and rage, and then suddenly, why do I have these giant claws growing out of my forearm? Guys, as you can see, illustrated by that picture over there, although far more melodramatically than I did. Hello.
Hello.
Okay, basically, after that, after a bunch of things, we don't go into because it's not relevant to the backstory. In this case, he was taken by Dr. Cornelius sometime after World War II, so he's much older than depicted, or at least much older than he wants people to believe. Sometime after that, he was taken to be part of the Weapons Expert program, and that involves finding this strange unknown magical element known as adamantium. No, it doesn't exist, to the skeleton. I will deliberately not be talking about adamantium to the skeleton because I don't think it has much relevance when I feel to study it, nor do I think it's possible at all. But there are some things that actually are kind of applicable in real life. It's a bit of a stretch, but I can link it to something, and that's called fibrodysplasia ossificans progressiva, or FOP, because it's way easier to say. FOP is a mutation of the gene ACVR1, and it prompts ossification, so calcification or turning into bone, of cells and tissues that are exposed to trauma, whether that be a blow or a cut or some kind of other wound. It's inherited at birth. A lot of ways people can know that their child has, because it usually doesn't display many symptoms at birth. It's not until you get trauma that this actually occurs, but the only way you can tell is that the foot, that's the big toe. That's what the big toe looks like, if that makes sense. So, does that make sense?
Yep.
Okay, but ACVR, the gene that's mutated, codes for a receptor protein that is on the same pathway as a bone morphogenic protein. And that receptor protein itself is also involved in signaling the body that the area that cell has been involved in trauma. The fact that it's on the same pathway means that some regulatory sequence has probably, it's been screwed. The regulatory system of that has been screwed, and as a result, as part of the cell healing process, cells start to calcify. And as you can see, here's a case study done by, I don't have the source, done by some scientists. Here's the patient, the same patient that, as you can see, as a result of exposure to force or whatever, bone, there's now bone between the ribs, lots of connective bone over here. The spine is completely fused, as well as the forearm here and the upper arm here, which is kind of scary to look at, but also kind of cool. Okay, as to super fast healing, I could spend like 3, 4, 7 lectures on trying to explain or think about how super fast healing could work, since there are so many genes involved in that process. Regulation for faster response times and more efficient pathways would be a huge effort, but one way I was thinking about on the way here was, and this is usually unlike humans, because we have a large amount of chromosomes, the genes tend to be spread out along all of them. So even if they're related, they'll just be all over the genome, which makes it less efficient to sort of find the gene that you want to code it into RNA, etc., etc. Whereas with bacteria, because it's one single giant loop of DNA, all of the related genes for a specific metabolic pathway will usually be in the same area. So you can just find the start of it and then code 4 genes in one hit. Bang, 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 bang. And that will usually, and that's like ludicrously more efficient. So I was thinking, it would have to be, and this doesn't even take into account healing time, just generation of proteins, but something like having a more efficient structure of where the genes may lie. However, so unrealistic. Very, very unrealistic. And I'm not going into animation because just no. Just no. Any questions or anything anyone would like to add? Ooh. The next one is Deadpool. I really liked Deadpool, even though there is not really any story on Deadpool, because that's kind of the wonder of Deadpool. He likes screwing everyone over and confusing people and breaking the fourth wall a lot. His backstory, as a result, is really, really up in the air. The current theory that seems to be going around is after he was diagnosed with a terminal cancer, he enrolled into Department K, which was a cross-country US and Canadian initiative to make superhumans. He was given an implant of healing factor, which I misspelled there, so healing factor. He was given a transfusion of this healing factor that was provided to Department K, and it was derived from Wolverine himself. So that halted and slowed down his cancer a lot. But it wasn't until way later where there was some kerfuffle in a prison and this sort of prison inmate cockfighting thing went on, and Deadpool decided no, he wanted to stop this sort of cockfighting.
So cockfighting or
well,
prison brawl.
It's the prison inmate equivalent of cockfighting, where higher ups will basically take prison inmates and say fight or die, or do both.
There's a different kind of cockfighting.
I will throw this at you. Okay. But yeah, basically what happened there is, I think the bigger reveal of his powers was basically he was stabbed through the heart and left for dead, and then somehow rose again. And because the name of this sort of organized fighting was called the Deadpool, that's how he got his name. Okay. So that's about all we know, because he himself keeps changing the story, and then the writers don't know, like there's a bit where they thought Loki was Deadpool's father. I'm not kidding, which is really strange. But yes, Deadpool's a very strange one, and I really like his character. Now, this image is very important, because I think Deadpool is a more accurate description of super fast healing. Because cells replicating and regenerating themselves at such speed would be very, very inefficient when it comes to error checking. DNA polymerase already has some correction mechanisms in place, but there can be quite a few mutations that slip under, especially when you take into account the number of human cells in the body. It may seem like such a tiny, insignificant number when you're just looking at a cell-by-cell basis on the likelihood of mutations, but because we have trillions and trillions of cells in us, it adds up fairly quickly. Now, Deadpool, when he has the mask off, looks There are several artist adaptations and depictions, but usually he's either covered in a ton of scar tissue or he's covered in a ton of tumors. And that's because, as I said, accumulation of mutations can often result in cancerous results. And as you can see, I have this wheel, and there's sort of how can I describe it? It's cancerous attributes. Well, attributes of cancerous cells that are basically perpetuating this self-proliferation. So I've got, you can't really see it, so you've got a sustained proliferative signaling, you've got to evade growth suppressors, you need to avoid destruction by the immune system, you need to enable replicative immortality, big word. You need, it's usually tumorous, so it'll promote inflammation, it'll have
metastasis.
Yeah, it will have metastasis, so it will spread. It will spread basically. It'll induce angiogenesis.
Blood vessels.
Yes. And the genome will be very unstable and mutated. It will also resist cell death, which is known as apoptosis. And deregulating cellular energetics. So basically, everything goes into overdrive. It doesn't die. It just likes replicating and replicating and replicating. And although super fast healing, I still have major problems with, this is probably the most acceptable version I will ever come across, because it is shown, and usually it's used as a metaphorical tool, but I like it how at certain points of the story you'll see Deadpool with more and more tumors on his body, or more and more scar tissue, and I kind of like it that we have an ugly superhero, because too many superheroes are big and busty have weird boob physics. But I also like Deadpool because he's freaking Deadpool and it's great. But yeah, as you can see in the previous one, this is Deadpool's face. And, sometimes it's worse, sometimes it's better. But I think it's probably the most accurate representation of super healing that has ever really happened in comics. Okay, the third case study, I think, is the most bizarre. Why did you move? Whatever. You saw him for a split second. Yay! Okay. Maggot's a weird one. I've only found out about him recently, but I just love his story, so I thought I'd include him. Okay. Maggot is also known as Japheth, and he ran away from his home in South Africa after discovering that his malnourishment was likely due to stomach cancer. He could only eat certain types of food, and it wasn't working. He was just wasting away. When he was stranded in the Kalahari Desert, he was found by Magneto, who actually helped him discover his condition. His digestive system was two techno-organic slugs living in the cavity that was his stomach and everything else. They could leave his body at will, actually eat their way out of his body, to consume and dissolve any substance it wanted at rapid speed, and then eat their way back into his stomach, filling the hole behind them and providing him nutrients.
What to say?
It is amazing. The geneticist in me is freaking out, but I love it. Okay, so, could this ever happen in real life? No. Before I continue, just think about that and try and imagine any case.
Like a
where
giant
it might
tapeworm.
happen.
Well,
Sounds like
tapeworm.
yeah.
Take one. Terrified.
Not the eating out and then eating back in.
Yeah, that bit's a bit interesting.
Are there certain animals that vault up their own in their gut?
Well, it's just having them do it at will and chewing their way out.
I also want to mention, and I wanted to wait until after you had this discussion for it, but their slugs can talk to maggots telepathically, and somehow, by consuming these objects, maggot has the intuitive knowledge and memory of that specific object. It just gets weirder and weirder, and basically it goes from, this is actually kind of cool and maybe plausible, to no, never was, no.
Objects have memory.
Apparently.
According to the X-Men universe, yes, which is supposed to be Earth, so no. Okay, my response to this is if you were to, yourself realize it's very, very hard to, which is why I obviously didn't come in the movie. Evidence suggests that mitochondria, which hang on, I'm gonna head over here. Mitochondria, which is these little things here, the sort of the energy factories of the human body. They take things and they make energy out of them. Evidence suggests that what happened was a protobacterium invaded the host cell. So here's an early eukaryotic cell. The proto-eukaryote decides it wants to infect the cell, which are these things here, and then really likes it. Hang on, no, here's the protobacterium. I'm getting things wrong. Protobacterium decides to stay there and actually really likes it. So it stays there for good, and whenever the cell replicates, the bacterium replicates on its own. And over millions and millions of years, it evolved into the mitochondria we have today. And the reason why we think that is because the mitochondrial DNA is completely different to our genome. And it's very, very highly conserved. And what I mean by that is it's very, very unlikely to change over time. It's not very susceptible to mutations at all. And so we have this theory called the mitochondrial Eve theory, where it was just one mitochondria that initially infected a cell, but as the mitochondria are passed through the maternal line, because the egg here, well, the sperm doesn't have any mitochondria. The sperm is literally just sort of a capsule of DNA plus what it needs to get to the egg, and that's it. This contains all the organelles plus half the DNA needed, if that makes sense. Yeah? So the mitochondria is passed along the maternal line. As the DNA is highly conserved and separate to the nucleus of DNA, to sort of our human quote-unquote DNA, we can sort of track it down to one organism known as the mitochondrial Eve, which is likely a protobacterium because of it's a plasmid. It's plasmid shaped. It's just one giant circle, which is evidenced in bacteria today. That's about as close as I can get to two giant slugs. And that's probably and I think that's okay. It's sort of a huge deviation from it. But as I was trying really hard to find something in nature apart from, that had evolved, that is a genetic kind of link to the human body and how it works, that was about as close as I can get. However, we also do rely heavily on bacteria for our digestion. Bacteria in our gut are crucial for breaking down food into nutrients, to create certain vitamins, to absorb all the nutrients while the intestines also absorbs all the water. I'll also note that I don't know the type of
Mitochondria.
Yeah.
Micro.
I've counted, I think I'm up to 10 typos.
I'm tired.
You're doing very well. Nothing wrong. Look.
Okay. I'll also note that animals probably rely on it a lot more than us because while we try and get most of our vitamins from the food directly, animals, particularly dogs and cats, or mostly carnivorous animals, usually have bacterium that produces chemicals like vitamin C in their gut for them instead of eating food that is rich in those sources. Can anyone think of any other representations of?
All I can think of the giant tapeworms. They
Yeah.
usually don't share nutrients.
Yeah,
They're not good at sharing.
I can't think of anything that's human.
It doesn't have to be humans.
We do have those, is it spiders? No, wasps that lay their eggs inside caterpillars and spiders, and they eventually eat their way out of them.
Yeah.
But they don't eat their way back in,
And they don't
share
need nutrients. Yeah.
Yeah.
It's a one-sided
Yeah, and
relationship.
there are, I think there's certain fish or gastropods, I think, that kind of I think it's sea cucumbers that eject their gut,
Yes.
grab something and then suck it back in.
Starfish do that too.
Yeah. So I think there's echinoderms
thing I can think of is there are fish that sort of basically live along with whales and things and just clean them off.
Yeah.
So things that clean out parasites rather than
So
provide nutrients.
evolving, sort of co-evolving together to have a sort of symbiosis.
Imagine if we had to spit our gut out to eat things.
I never want to go on dinner dates ever.
Wikipedia refers
Don't say sea cucumber.
a quick search on the interwebs reveals the superglided tube worms.
Yeah.
So the tube worms and symbiotic bacteria live at hydrothermal vents in the ocean floor.
Oh, nice.
So these worms have no digestive tracts at all. And so they are wholly reliant on their symbionts for nutrition. The bacteria oxidize hydrosulfate methane, which the host applies to them. And they supply nutrients.
That's pretty cool.
That's pretty cool.
Also, rhizobacteria in plants, the bacteria that live in plants and fix nitrogen for them, and like rely on the plant for support and for other nutrients, but also fix nitrogen for the plants, which is cool.
So yeah, there are a lot of examples of similar things happening in nature. My list obviously isn't exhaustive, but it was the only one I could really find that is sort of permanently in the human body and not a sort of co-evolved symbiosis, because of course Maggie is only one person. We don't all have techno-organic slugs digesting meals for us. That would either be really scary or really cool, come to think of it, but of course Maggie's only one person. I also want to add, because I forgot to last time, he named his slugs. He named one Amy and one Mimi. And that is
my god.
I know, Amy and Mimi.
This badass villain, I'm assuming.
No, he was part of the X-Men.
Oh, this badass X-Man.
He was
They
the
just like
youngest.
That's the cutest thing I've ever heard.
Yeah, he was the youngest X-Men, and then he died because they killed off the cool one.
Oh, rude.
I know.
Road.
Okay. So that's it for my little talk. I'm sorry the list wasn't the most exhaustive thing, but I kind of just liked looking at the X-Men universe and seeing what I could make of it, and seeing if anything was similar to the real world now. Thanks. Does anyone have any questions or comments before I'm completely done?
Do you think that perhaps radioactive spiders might have a higher success rate than genetic guns?
Jingoes have a pretty low success rate.
They do. They also probably wouldn't be able to get to veins being tilted just at the surface.
We also don't have a very high source sample for
If you were transforming an entire organism, I think radioactive spiders might be more useful.
Also,
your transformation is more like a transformation
but probably not the way you want them to, because unless they're biting and injecting venom laced with nanoparticles, you won't specifically get the mutation you're out. You want to engineer it?
Yes, I really do. You want fun?
But yeah, radiation is one of the nutrients that was on the slide earlier, where if you wanted to mutate things randomly, you'd probably expose them to radiation, or UV radiation, expose them to chemicals that would promote mutations on the cell surface. A radioactive spider, even by walking on someone, would probably cause some change, whether or not it's noticeable or not, that's another issue entirely. Whether or not it's what you want, that's an entirely different kettle of fish, but I mean, that'd probably be more effective than a gene gun.
Well, if a gene gun, then we should get at least 7.
Maybe.
But I think it'd be more successful causing mutations, but gene guns are more specific.
Yeah,
But like,
That's what you get to choose.
when you shoot a gene gun at a petri dish, the center of it is destroyed.
Yeah, like it gets like the reason it doesn't transform like a billion of them is because they die.
Yeah. And it just sucks for the rest of the people, but there'd be like 7.
Just shoot it from the moon and
Nothing can go wrong.
there'd be at least 7 survivors.
Well, the nanoparticles are usually made of gold, so that's gonna cost.
Gold or platinum. So yeah, gene guns are not only not very effective, but they're also very expensive.
That idea of
virus poison, that just reminded me, there are a number of creatures, especially Komodo dragons from memory, that are not, don't have a venom of their own, but their bite is effectively venomous because they've got so many bacteria in their mouths that you still end up with massive necrosis of the flesh. So that could almost be akin to that idea of sending out something to pre-digest before slurping it back in.
Don't spiders dissolve poison where they injected that?
It dissolves them into
Yeah, they squirt out the digestive juices into the little wrapped up package of glory until it liquefies and they go.
most effective way to accept.
How do you spell?
S
With
C
extra
L U U U U U U U R.
Peace.
There's also, I think most big poison dart folks that if you raise them entirely in captivity, they won't be poisonous because they need to take something from the environment to become toxic.
We don't know what it is, but we know it's something.
But I wouldn't be surprised. Nature's
For some of
really
the
toxic.
species.
There's also that one guy who injects himself with snake venom thinking it'll make himself stronger.
Yeah, then he needs his life just be injected the wrong strain one day.
It's much more likely to just make you allergic to the anti-venom.
And he makes himself he makes cocktails of snake venom for himself.
Well, if you drink it, it's fine. Snake
He
venom.
injects it into
Oh,
himself.
that's dumb.
Yeah, some people are idiots.
Some people should be allowed to do their experiments.
But
I think we should
Darwinism needs to take form eventually.
But snake venom's a really cool venom in that sense, in that because the snake has to eat what it bites. If you swallow snake venom, it's not in the least bit poisonous. It's only if it's actually injected into your bloodstream that it becomes a problem, or in some instances if it gets on your skin.
It coagulates. A lot of blood snake venom works in that it turns your blood into jelly.
Mm.
So essentially what you have is
Jellified meat.
yeah.
Jellied eels, anyone?
There's a picture on YouTube of someone putting a drop of snake venom into a shot glass full of blood and then they tip it upside down and it wobbles.
Has anyone started making cocktails, drinking cocktails with snake venom as a flavoring?
Back a couple of thousand years, yeah. Nowadays the therapeutic goods.
You can still get it in sake.
It sounds like
yeah.
the sort of thing that the delicacy somewhere.
No, in Japan they have the sake which has snakes in them, and occasionally they add some of the venom.
There are an awful lot of either prehistoric or early historic societies that make a big thing of drinking venom, mostly because you do that with snake venom, because if you're not doing it with snake venom, you're dead. But nowadays the Therapeutic Goods Administration starts asking a lot of awkward questions when you're trying to sell people poison.
Only in Australia.
True, in America it's the Food and Drugs Administration asking a lot of awkward questions when you're trying to sell people poison.
I'm just asking if you can go to random, barely developed or developing tourist places.
I'm sure Russia can sell you a great many things.
Russia, you can get anything you want in Russia.
Any more questions, comments, queries, anything?
One comment. The
Yeah.
mitochondrial Eve thing is something else. That we can trace our lineage back to this one. It was a woman about 200,000 years ago.
Okay,
Rather than a
Yeah.
bacteria.
It can't be 10,000 because we're a bit too far spread out at that point.
There's a lot of
Problems with that theory in particular.
Yeah.
Zach Master's theory is that it's 10,000 years ago, but whether or not that is.
Probably because
Well, I just
if you
verified
were to
online.
go any further,
Well,
you
I
would be
remember
unlikely to
mentioning
find genetic matter.
When you have enough complexity, sometimes it's possible there were more and they just kind of got spread out randomly.